Nucleic acids are made of monomers called nucleotides. Each nucleotide consists of a nitrogenous base, a pentose sugar, and a phosphate group. The portion of a nucleotide without the phosphate group is called a nucleoside, with only a base and a sugar. There are two families of nitrogenous bases, or nucleobases. Purines have a 6-member ring fused to a 5-member ring. Purines include hypoxanthine, which has a ketone group on the 6th carbon. The nucleoside of hypoxanthine is called inosine, and the nucleotide is called inosylate. Guanine also has a ketone group on carbon 6, as well as an amine group on carbon 2. Its nucleoside is known as guanosine, and its nucleotide is known as guanylate. Adenine has an amine group on carbon 6. Its nucleoside is known as adenosine. The most important high-energy compound in biochemistry is ATP, which stands for adenosine triphosphate, which is adenosine plus 3 phosphate groups. The nucleotide of adenine is known as adenylate. Pyrimidines have a single 6-member ring. Uracil has ketone group on carbon 2 and carbon 4. Its nucleoside is known as uridine, and its nucleotide is known as uridylate. Thymine has a very similar structure to uracil, with the addition of a methyl group on carbon 5. The nucleoside of thymine is known as thymidine, and the nucleotide is known as thymidylate. Cytosine has a ketone group on carbon 2 and an amino group on carbon 4. Its nucleoside is known as cytidine, and its nucleotide is known as cytidylate. A DNA molecule has 2 polynucleotides spiraling around an imaginary axis, forming a double helix. The 2 backbones run in opposite 5' to 3' directions from each other, an arrangement referred to as antiparallel. The sequence of bases along a DNA or mRNA polymer is unique for each gene. The nitrogenous bases in DNA pair up and form hydrogen bonds. In DNA, adenine base pair with thymine through 2 hydrogen bonds, and guanine base pair with cytosine with 3 hydrogen bonds. Therefore, GC base pairing is stronger than AT base pairing. Adjacent nucleotides are joined by phosphodiester bonds that form between the hydroxyl group on the 3' carbon of one nucleotide and the phosphate on the 5' carbon on the next. These links create a backbone of sugar-phosphate units with nitrogenous bases as appendages. The DNA double helix contains a major groove and a minor groove. The major groove occurs where the backbones are far apart, and the minor groove occurs where they are close together. The grooves twist around the molecule on opposite sides. There are 2 main aspects of structural variation in DNA. The ring of sugar in DNA is actually not in a single plane. It is puckered. The sugar puckers in DNA structures are predominantly in either carbon 3' endo, in which carbon 3 points in the same direction as carbon 5, as well as carbon 2' endo, in which carbon 2 points in the same direction as carbon 5. The second aspect of structural variation has to do with the rotation about the glycosylic bond between the nucleobase and the sugar. The anti-conformation has smaller group above the sugar ring, while the syn conformation has a larger group above the sugar ring. Due to steric hindrance, the anti-conformation predominate in nature. DNA molecules have 3 structural variants. The B form is the most stable structure for a random sequence DNA molecule under physiological conditions, and is therefore the standard point of reference in any study of properties of DNA. The A DNA is widest in terms of diameter. It is a right-handed helix with glycosylic bond in anti-conformation and sugar pucker in 3' endo conformation. It has 11 base pairs per helical turn with 2.6 angstrom, which is 10 to the negative 10 meters, helix rise per base pair. It has a relatively deep major groove and relatively shallow minor groove. The A DNA form exists in both DNA and RNA. The B form DNA is also right-handed and has a diameter of medium width. Its glycosylic bond occurs in anti-conformation and has a sugar pucker of 2' endo. It has 10.5 base pair per turn with 3.4 angstrom of helical rise per base pair. Its major groove and minor groove are standard in size, and it is the primary form of double-stranded DNA. Z-form DNA is narrow in shape, and it is a left-handed helix which is different from both A DNA and B DNA. Its pyrimidines are similar to B DNA with anti-glycosyl conformation and 2' endo sugar puckers. However, its pairings occur in syn-glycosyl conformation and has 3' endo sugar pucker. It has 12 base pair per turn with 3.7 angstrom of helical rise per base pair. It has a shallow major groove with deep minor groove. The DNA backbone of a zDNA occurs in zigzag appearance, and the zDNA may play a role in regulating the expression of some genes or in genetic recombination. Double-stranded DNA can be denatured to single-stranded DNA with increase of temperature. Tm, or the melting temperature, is the temperature at which 50% of DNA has been denatured, and it represents the stability of double-stranded DNA. The melting temperature of a DNA is directly proportional to the percent of guanine and cytosine base pairs. Recall that guanine and cytosine base pairs have 3 hydrogen bonds, which is more stable than adenine and thymine base pairs. The melting temperature also increases with lower pH and higher salt concentration. Double-stranded DNA can be denatured by urea and formaldehyde, which disrupts the hydrogen bonds. DNA denaturation can be detected by monitoring UV absorption at 260 nanometers. The hyperchromic effect refers to a striking increase in absorbance of DNA upon denaturation. As double-stranded DNA denatures, the interaction between bases are reduced. More bases are in free form, therefore UV absorbance of DNA increases. Certain DNA sequences adopt unusual structures known as repeat structures. A palindrome is a phrase that is spelled identically in either direction. The term is applied to regions of DNA with inverted repeats of base sequence, having twofold symmetry over 2 strands of DNA. They are self-complementary within each strand and have the potential to form either hairpin or cruciform. A mirror repeat is an inverted repeat that occurs within each individual strand of DNA. Mirror repeats do not have complementary sequences within the same strand. Therefore, they can't form hairpin or cruciform. It is found in intramolecular triple-stranded DNA, or HDNA. The H stands for Hustine pairing, which is an unconventional pairing that allows a cytidine residue to pair with the guanosine residue of a GC base pair, and a thymidine residue to pair with the adenosine of an AT base pair, allowing the formation of a triplex DNA. 4 DNA strands can also pair to form a tetraplex, which only occurs readily for DNA sequences with a very high proportion of guanosine residues, such as in telomere repeats of TTAGGG. There are 3 important types of DNA repeat sequences in E. coli. The methylation of adenine of the sequence Ch.8 in newly synthesized DNA plays an important role in the regulation of replication, as well as recognition of the newly synthesized strand in mismatch repair. The palindromic sequence G.8 is cleaved by the restriction endonuclease EcoR1 as a defense mechanism against invading viruses, while the host DNA is protected by a modification enzyme that blocks the cleavage. The formation of hairpin structure plays a huge role in the role-dependent termination of transcription, as well as attenuation in the trip operon. The Wobble base pair refers to pairing between 2 nucleotides in RNA molecule that does not follow Watson-Crick base pair rules. It is due to the fact that the 5' position of the tRNA anticodon can often base pair with different nucleotides on the 3' position of the mRNA codon. Uracil can base pair with both adenine and guanine. Guanine can base pair with both uracil and cytosine, and hypoxanthine can base pair with uracil, adenine, and cytosine. The most stable forms of nucleobases occur in amino and keto forms. Amino form of adenine can base pair with keto form of thymine and uracil, and amino form of cytosine can base pair with keto form of guanine. Lastly, DNA and RNA differ by the 2' carbon. DNA stands for deoxyribose nucleic acid. It has a hydrogen atom on the 2' carbon, whereas RNA stands for ribose nucleic acid and has a hydroxyl group on the 2' carbon. Single-stranded DNA is 330 Daltons, and single-stranded RNA is 345 Daltons. DNAs contain thymine, whereas RNAs contain uracil.